Hi everyone, in this video I will explain the chemistry of sphingolipids in detail. It's a continuation of our last class, that is chemistry of phospholipids. So phospholipid we are classifying into two on the basis of the type of alcohol group. Glycerophospholipid, which has glycerol as the alcohol part, and in sphingophospholipid the alcohol part is contributed by an amino alcohol, that is sphingosine. And in glycerophospholipid, the basic structural unit is phosphatidic acid, but in sphingolipid, the basic structural unit is ceramide. So, what is the structure of a ceramide? Ceramide is formed by the esterification of sphingosine with fatty acid. So, ceramide composition is sphingosine with fatty acid. This is the basic structural unit of sphingolipids. And the sphingolipids are again subclassified into two. One is phosphorylated sphingolipids and the second group is non-phosphorylated sphingolipids. And for phosphorylated sphingolipid, it is also known as sphingophospholipid. The example is sphingomyelin. And for non-phosphorylated sphingolipid, the example is glycolipid. So sphingomyelin is phosphorylated sphingolipid and glycolipid is non-phosphorylated sphingolipid. So there are two types of sphingolipids depending on the presence or absence of phosphoric acid. Phosphorylated sphingolipids, that is sphingophospholipid, and non-phosphorylated sphingolipids. And sphingomyelin, it is the only one sphingolipid which is phosphorylated. So you should have some idea regarding the structure of sphingolipid. In sphingomyelin, it is a type of sphingolipid, so the basic structural unit is ceramide. So what is ceramide? Sphingosine with fatty acid, that is the ceramide. And in sphingomyelin, they contain some additional group, that is phosphoric acid and a nitrogenous base, that is choline. So sphingomyelin contain sphingosine, fatty acid, phosphoric acid and a nitrogenous base, that is choline. So ceramide, phosphoric acid and choline, this is the structural composition of sphingomyelin. So regarding sphingomyelin, it is a type of phospholipid. It does not contain glycerol, instead they contain an amino alcohol, that is sphingosine, and it is predominantly present in neuronal tissue, and they does not have carbohydrate residue. So phosphorylated sphingolipid does not have carbohydrate residue, but non-phosphorylated sphingolipid, they contain carbohydrate instead of phosphoric acid. And sphingomyelin is the only sphingolipid which is phosphorylated. Coming to the second type of sphingolipid, that is non-phosphorylated sphingolipid. It is also known as glycolipid or glycosphingolipid. The reason is they contain carbohydrate instead of phosphoric acid. So glycolipids are phospholipids that contain carbohydrate without having phosphoric acid. And the examples are cerebrocyte, globocyte and ganglioside. So all these glycolipids contain carbohydrate. So what are the difference? In cerebrocyte, the carbohydrate part is contributed by monosaccharide residue. So if it is a glucose, then it is known as glucocerebrocyte. If it is a galactose as the monosaccharide residue, then it is known as galactocerebrocyte. So for cerebrocyte, the examples are glucocerebrocyte and galactocerebrocyte. And cerebrocyte contain monosaccharide residue along with ceramide. So what is a globocyte? Globocyte contain oligosaccharide residues instead of monosaccharide. So this is the structural composition of globocyte, that is ceramide and oligosaccharide. Ceramide is the basic structural requirement for sphingolipid. So all sphingolipid contain ceramide unit. So in globocyte they have oligosaccharide along with ceramide. And what is the ganglioside? Ganglioside structure is almost similar to globocyte, but they have some additional sialic acid residue. Here the sialic acid residue is N-acetyl neuraminic acid. So this is the structural composition of ganglioside. They have ceramide and they have oligosaccharide residue and they have additional sialic acid residue, that is N-acetyl neuraminic acid. So what is a sialic acid? It is a monosaccharide having 9 carbon atoms, that is sialic acid. and N-acetylneuramic acid is a type of sialic acid which is present in human tissues. So that's all regarding the classification of sphingolipids. There is one short answer question, that is plasmalogen. So what is the structure of plasmalogen? They contain glycerol unit, and in glycerol unit, the hydroxyl group of the first carbon, it is attached with an unsaturated hydrocarbon chain. So instead of ester linkage, they have ether linkage. So the first carbon of the glycerol unit have an ether linkage. Hence, this plasmalogen is also known as ether lipid. And these plasmalogens are predominantly present in nervous tissues. So this is the classification of phospholipid. We can classify phospholipid into two on the basis of the alcohol part. In glycerophospholipid, the alcohol part is contributed by glycerol, and in sphingophospholipid, the alcohol part is contributed by sphingosine. And this glycerophospholipid is again subclassified into nitrogenous glycerophospholipid and non-nitrogenous glycerophospholipid. So nitrogenous glycerophospholipid contains some nitrogenous bases. For example, phosphatidyl contain choline, phosphatidyl contain ethanolamine as the nitrogenous base, and phosphatidyl contain serine as the nitrogenous base. And phosphatidyl inositol and cardiolipin. These are the examples for non-nitrogenous glycerophospholipid. Coming to sphingolipid subclassification, it is subclassified into phosphorylated sphingolipid and non-phosphorylated sphingolipid. Phosphorylated sphingolipid is also known as sphingophospholipid, and the example is sphingomyelin. Non-phosphorylated sphingolipids are known as glycolipid, and the examples are cerebroside, and they have monosaccharide residues, and globoside, they have oligosaccharide residue, and ganglioside, they have oligosaccharide residues with sialic acid component. So that's all regarding the chemistry of sphingolipid. Thank you.